Right, welcome. We are going to start talking about genetics this evening. We've already kind of done some precursors to this earlier. Um, so we're going to continue on with some of the more specifics as we go through. Yes, this is a great attention grabber because everyone loves a cute puppy. So we're going to first start talking about Gregor Mendel. Gregor Mendel, we're going to talk about who he was and what he did. He's kind of the first geneticist, if you will, to give us a basic understanding of what genetics were. Most of our understanding of what heredity is, or the process of transferring traits from parent to offspring, started with him and his experiments. Mendel was a scientist and a monk who was trying to find a pattern of probability in heredity. So if you remember from your algebra classes when you dealt with probabilities, we're going to be touching on some of those in this section. So just remember, probabilities is the part of the whole, and that will get you quite a ways. So the advantage of studying the pea plants that he used was that he could control the matings. And we've already talked about plant reproduction, so you could see how he could control the matings by controlling the anther and controlling the stamens and the pistils and things like that. So he could control those, so having an understanding of that comes in kind of important. Mendel focused on studying the inheritance of 7 characters, and those characters had to do with the seeds. They could either be round or wrinkled, they could be yellow or green. With the flowers, they could be white or violet. The pods, the form and the color of the pods, and they were either full or constricted, or they were colored, they were yellow or green. And then he studied the stem, the place of the stem and the size of the stem. Did they have axial pods or terminal pods? Axial pods means they have pods down here on each axis. Terminal pods mean they only bloom out here at the very ends or the tips. And the stem size, were they tall or were they short? So those are the 7 characters. So the seed form, the seed color, the flower color, the pod form, and the pod color, and then the stem size and the stem place. So each character has 2 traits or variations. So that was key as well. There weren't any other options. They were either this or this. So we'll talk about what that means with inheritance a little bit later. So what he would do is he would cut the pollen off of the anthers and the stamens of his experimental plants, and then he would brush or introduce the pollen from another plant. So if you remember, he would cut off here the anther, and he would then use his own up here in the stigma. So he would cut off the anther and the stamen, and he would put pollen from a different plant up here and brush it on to the stigma. So therefore, the reproduction was a cross-pollination, not a self-pollination, if you remember those terms. This allowed him to be completely sure of the parents of each of the new pea plants. It's a very, very controlled experiment. So, in one experiment, Mendel selected the purple and white flowered pea plants to cross. So he's crossing just the flower color of those 7 traits here. The plants were true breeding, which meant they all produced the same color when they were self-fertilized. So whenever the purples self-fertilized, they produced all purple. When the white self-fertilized, they produced all white. So they were true breeding. That means both of the parents were purple or both of the parents were white flowered colored. True breeding parents in experiments like this are called the pea generation. The pea generation is that one that is, think of it as pure generation, okay, or that first generation. So the pea generation, they're pure, and they have all purple or all white flowers, and their parents were both of that. So they are pure purple or pure white. The hybrid offspring in this monohybrid cross are called the F1. They consisted of all purple flowers, okay? But they are not pure, even though they're purple. They're F1s because they are hybrids, because we know we crossed a purple and a white flower. So that's that F1 generation. A monohybrid cross involves parents that differ in one trait only. So for thinking about F1 generations, those are the generations of hybrids. So when the individuals from the F1 generation were crossed, resulting in the F2 generation, they had a predictable ratio of colors. So we had our pure generation, we took our F1s, and we crossed two F1s, okay? So we had an F1 and an F1 here. They were both purple, but in the F2 generation, we have a 3-to-1 ratio because we have 3 purples and 1 white. So he had 705. He did a total of 929 flowers, but about 705 of those 929 were purple. So his experiment size was pretty large. So he had 75%, or that is a ratio of about 3-to-1, because 224 were white. It's about 25%. These same results occurred with the 6 other pea plant characteristics he studied. So this occurred over all 7 of the traits he was studying for. He got a 3-to-1 ratio. So therefore, his 4 hypotheses were: there are alternative versions of genes that account for different traits within a character. We call those alleles, A-L-L-E-L-S, alleles. They are called alleles. I guess I got ahead of myself. For each character, an organism inherits one allele from each parent. If the alleles differ, only the dominant allele will be expressed. The recessive allele has no noticeable effect. A sperm or egg will only carry one allele for an inherited character due to the segregation of the chromosome pairs during meiosis. Now, that one gets a little more complicated there. Okay, so a sperm or egg will only carry one allele. So the sperm carries one, the egg carries one. And this is the inherited character during that segregation. So during meiosis, they're only able to carry one of the alleles. Okay? The recessive one didn't have an effect yet. This is called the law of segregation. Okay? These are his 4 hypotheses. Keep note of them. They are on the study guide. So, what the possible combinations and probabilities of a monohybrid cross can be shown with a Punnett square. You probably haven't seen these since high school, but here we go. So, what we have is the male pollen and the female pistil. So that's the way we do it. The 2 possible alleles from the father are shown along the top. So we always put the father on the top, the mother on the side. Okay? And the 2 possible alleles from the mother are shown along the bottom, or essentially the side here. The capital letters represent dominant alleles. So dominant is the big B here and a big B here. Now, you could do big B and little b, because whenever you're doing a monohybrid, you always keep the same letter, just the lower case of it represents the recessive allele. So keep that in mind. Lowercase letters represent recessive alleles. You don't want to change the letters. You want to keep one letter for one trait. So you wouldn't want to put it wouldn't work if you put a P, capital P, and a lowercase w for purple and white. It wouldn't work. Okay? You need to have the big B, little b, because science says so. All right. So each box of the Punnett square shows a different genotype. The genotype are the genetics of that organism. You can't see genetics. You can't see the genotype. What we can see is the phenotype, which is the color or the expression of that. 25% are homozygous dominant, or BB. That's this guy right here. It is homozygous dominant. So it got both. It got a purple allele and a purple allele from its father and from its mother. 50% are heterozygous. That's these two. They got from their mother here. They got the purple. From his father, he got the white allele. He, she, it, plants don't have sexes, I guess. But this one, then from the father, got the dominant purple and the recessive white. Then 25% are homozygous for the lowercase b's. So from the father, he got the white allele, and from the mother, they got the white allele. So it's expressed as 2 lowercase b's, or the white. The phenotype is the actual physical result of those traits that we see. Okay, so this is represented as the genotype, the big B, little b. The phenotype is represented by purple, 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 white. Okay, 75% purple, 25% white. So when we do the phenotypes, there is a ratio of 3 to 1. If you do the genotypes, it's 1 to 1. Keep that in mind. Okay? Same thing goes when we have a homozygous dominant or heterozygous offspring with the test crosses. Okay? So now we have a homozygous dominant, which is homozygous means the same. And we have a heterozygous offspring over here, meaning it has one of each allele. This has the same allele. So the only difference is the genotype by conducting this test cross. The unknown genotype is crossed with a homozygous recessive individual. So in this case, we have the phenotype of a homozygous dominant and a heterozygous offspring are going to look identical. So this, I spoke incorrectly. This is a homozygous recessive individual, 2 lowercase y's. So we need to know if this individual is homozygous dominant or heterozygous. Okay, they could, if it was homozygous dominant, it would have capital Y here. But since it has capital Y, lowercase y, we're going to look at what the offspring are. So it gets the mother here gives the capital Y, father gives the lowercase y. That means it's yellow. Same here. Mother gives this one, father gives this one, so it's still going to be yellow. But then down here, the mother gives the recessive and the father gives the recessive. That means that it is going to show up with the recessive trait. Same thing in this case. That tells us that the mother, or our unknown, was actually a heterozygous individual. So we know she wasn't the part of the P generation. That was an F1 generation here. If the unknown individual is homozygous dominant, all of the offspring would show the dominant characteristic. So if she would have been homozygous dominant, you would have had this offspring for all of them. They would have had a large Y and lowercase y. And you would have had all yellow pea pods in that test cross. If the unknown individual is heterozygous, half of the offspring will show the dominant trait, the other half show the recessive, which is what we have here. A dihybrid cross. So now we're getting a little more complex. We're having 2 traits. So he set up a dihybrid cross where the first mated true breeding parent varieties had 2 different traits. So a dihybrid cross has 2 different traits. He crossed a plant with yellow and round seeds with one that had green wrinkled seeds, creating an offspring of all yellow and round seeds. We're going to draw this out whenever we look in class tonight. Okay. Second, he mated 2 of the heterozygous F1 plants, which all ended up this. So capital Y, lowercase y, big R, little r. He mated 2 of those plants. And this is what happened. So, again, the father is up here. So he has some that are yellow and round, some that are homozygous yellow and round, some that are homozygous yellow and heterozygous round, some that are homozygous recessive, so they're going to have the green, and those that are homozygous recessive are paired with the homozygous round. Then this one is considered heterozygous recessive for both traits. Okay, same thing going down this side. So then you just have to basically create a multiplication table chart here. So you come up with one that is homozygous and round. You come up with, this is homozygous yellow, heterozygous round. Heterozygous yellow, homozygous round. And you have then heterozygous yellow, heterozygous round. So you have, and you could go through all of these. If they have 2 capital Y's, they are homozygous yellow. If they have 2 capital R's, they're homozygous round. If they have big Y, little y, they're heterozygous yellow. If they have big R, little r, they're heterozygous round. If they have little y, they are homozygous recessive or green, because that's the recessive trait. You remember yellow is dominant and round is dominant. If they have 2 little r's, they are homozygous recessive, or they have the wrinkles. Okay, we will go through this again in class, and I'll draw it out. The resulting F2 generation showed a 9:3:3:1 ratio of possible phenotype combinations. So we have 9 that were yellow and round. 1, 2, 3, 4, 5, 6, 7, 8, and 9. We have 3 that were yellow and wrinkled. 1, 2, and 3. We have 3 that were green and round. 1, 2, 3. And then we have 1 that was completely homozygous recessive, which was green and wrinkled. So these, of all of these, there are only 2 that are homozygous. This one is homozygous dominant. This one is homozygous recessive. The others are all heterozygous. These traits were inherited from genes on different chromosomes and did not affect each other's inheritance. They were not linked together. This is called the law of independent assortment, when those genes are not linked together. He then started studying some other things that dealt with other issues like incomplete dominance. Incomplete dominance means not all the traits follow the simple dominant and recessive patterns. When true breeding red and white snapdragons are crossed, the resulting F1 generation are all pink hybrids. They're all heterozygous. That means there's no dominant and recessive trait. There's incomplete dominance, because the hybrids have a phenotype somewhere in between the true breeding parents. Codominance. Some genes have more than 2 alleles within the population. The primary blood types in humans are a result of 3 alleles that can cause certain carbohydrates to appear on the surfaces of their blood cells. A is dominant and B is dominant. O is recessive, so we have codominance here. An individual with A and B alleles will express both carbohydrates on their membranes. So maybe you have a parent that's, one parent is A, one parent is B, therefore you're going to be AB. So this is codominance, and both alleles are dominant and expressed. If you have one parent that's A and one that's O, you're going to have A blood. But it just depends if your and what your parents are. If your parent is like A with an O recessive or B with an O recessive, there could be something else in there that gets a little more complex. An individual with only O alleles will not express any carbohydrates, so they have no carbohydrate markers on the cell membranes. So, a heterozygous individual with both A and B alleles will express both types of carbohydrates on the membrane of their red blood cells. So type A has green spikes, type B has blue circles. If you are AB, you have both. If you are O, you have none. Type AB, individuals with two of the same dominant alleles or heterozygous with one allele O will express only one of the carbohydrates. So they're all A dominant, B dominant, or AO, BO. Okay? Type A, which is AA or AO. Type B, which is BB or BO. A type O individual must have two recessive alleles. So maybe you have a parent that's AO and a parent that's BO. You could, per the law of independent assortment, end up with O-type blood. You could also end up with AB blood. I want to be clear on that. But if you have a parent that's AA and a parent that's BB, you're going to end up AB. If you have a parent that's AO and a parent that is O only, you're going to end up, if you have a parent that's AA and you are A, and another parent that's AO, you could end up A or you could end up AO. So there's a lot of different types that you could end up with in there because of that independent assortment. Now we have polygenic inheritance. Some characteristics are polygenic and vary across the entire spectrum within a population because they're influenced by multiple genes. Those are a lot harder to track. Human skin color is controlled by 3 genes. Each gene has 2 alleles, one for darker skin and one for lighter skin. So this is where we get these independent assortments. So in the number of alleles that show up for each of these different ones, the majority of them are still in the middle. Then you have a chance of being closer in, and then you have a little bit chance. So the skin tone, if you have all 6 dark skin alleles, you're going to have a darker skin tone. An individual with all 6 light alleles will be at the other end. And individuals who fall somewhere in the middle of the spectrum are going to be the majority. So when you look at those numbers. There's a chromosome theory of inheritance. This means that each gene occupies a specific loci. A loci is a position on a chromosome. Both chromosomes undergo independent assortment during meiosis. It is during this time that different possible alleles within gametes are formed. This is the chromosome theory of inheritance. So, this happens during the P generation, and as we go down through here. So let's kind of take a look. In pea plants, seed color and texture are often different on different chromosomes. So there's an equal chance that each allele of making it to a gamete. So we have over here a homozygous yellow round. Over here we have a homozygous green wrinkled. So this one's dominant, this one's recessive. So we pass along the round yellow, we pass along the green wrinkled. So all F1 plants are going to be yellow and round, but we have some recession in here at this point because one parent had that. So then the law of segregation says we have 2 probable arrangements of chromosomes at metaphase. We can have these arrangements. We can have round and green, or we can have yellow. So we can, these are going to be, this is where it gets difficult. So here is where they are segregating out, and they're assorting themselves. They can either be, and this is all genotype. We don't have expressions of the traits yet. So then they go through, so they're going through metaphase where they're lining up in the middle, and we start to pull apart during anaphase. And we're pulling the round yellow and the green wrinkled out. So what we then end up with is as those pull apart, we end up with round and yellow. These pull apart, we end up with wrinkled and green. We end up with yellow wrinkled, and we end up with round yellow. So through these, we end up with these characteristics, or these gametes here. We have a yellow and round gamete. We have a fourth of them that will end up with our green wrinkled. We have our yellow wrinkled, and we have our green wrinkled. I may have said this one wrong. This one should be, oh, wait, this one is green round. So that one's going to be over here. This is green round. This is yellow wrinkled. This is our green wrinkled here in our F3 generation. Genes on the same chromosome tend to be inherited together, are called linked genes. Linked genes do not follow the law of independent assortment. There were fruit fly experiments done by Thomas Morgan Hunt, and he studied the chromosomal basis of counting fruit. Fruit flies bred at a very high rate, or every 2 weeks, and only have 4 pairs of chromosomes. They were ideal test subjects. He studied the body color and the wing size for linked traits to be inherited together. So he had, in his experiment, these were linked traits. So body size and wing type were linked together. So it got a little more difficult with it. So when you kind of study this, he performed between heterozygous gray-bodied flies with normal-shaped wings and homozygous recessive flies, black bodies and small wings. So you can see it gets a little more difficult. Most of the offspring, or 83%, had the same phenotypes as the parents. So that made it a little more difficult to kind of understand that they were linked together. A smaller percentage of the offspring, 17, had phenotypes that did not match either of the parents. These were called recombinant types, meaning they recombined and linked together. So it was a little bit different with that. He explained them as there should have not been possibilities unless the alleles were there. But it kind of happened. So, Scientists later discovered that this was due to crossing over taking place in meiosis 1. Remember, we talked about that, how traits will cross from one to another, and this crossing over took place. Alleles present on the sex chromosomes X and Y in humans take a unique pattern of inheritance called sex-linked traits. In females, sex-linked traits follow the pattern of simple dominance. XC is normal, XC with an X lowercase c is normal, X lowercase is red-green colorblind. That means they got the colorblind gene that's an X-linked from their mother. In males, the Y chromosomes are shorter and does not contain an allele. XCY is normal, X lowercase cy is red-green colorblind. So when we look at some pedigree charts, when studying sex-linked traits, it's helpful to look at these pedigree charts. Males are inherited by boxes, females are circles. So, as I was saying before I stopped, there are pedigree charts, and when we study these sex-linked traits, it's a lot easier to set up a pedigree chart. Males are by boxes, females are by circles. Affected individuals are colored in, not affected individuals are left blank. So you can see the mother is heterozygous and is a carrier. And if they are a carrier, they're shown as half-colored in. So only males can be full-on homozygous with it. So we can see that we have some problems here with whatever the trait may be, that some of them are carriers and some of them end up having the sex-linked trait. So one of the most famous pedigree charts helped to determine the carriers of hemophilia in the English royal family. So as you look through all of these, you can have carrier females. So hemophilia is carried by females and and manifests itself in the males. So we were looking at just this section of it earlier, but you can see how it goes through here. And, just all of these we go over here. This family, none of them had it. But Alice, the daughter here, did have it. So Leopold, one of their sons, had it. Beatrice, one of their daughters, was a carrier. So you can see how it goes down through and ends up and where it ends up. So just kind of showing you, this is the English royal family of today. And this is the English royal family way back when with Victoria and Albert. So this is part of the current British royal family. All right, that is all we have for this. I will see you in class.